Okay, let's start then. There's a very quick crash course of a total revision of class 10 biology portions. So we are expecting few more students to join by the time let us start. The biology syllabus itself is a huge syllabus and we have a limited number of days. So I would like to cover up two major portions today, that is nutrition and respiration. It's a quick revision. So I will be going very fast. So once you go through it, then let me know. We shall go through some of the question answer sessions. Okay. The first portions related to this chapter is about nutrition. Okay, so nutrition is actually both in plant as well as in animal. Let us start with a quick revision through the plants. So the few portion points are noted down. The life processes, there are four major portions in this chapter, that is nutrition, respiration, transportation, and excretion. And coming to the topic nutrition, we have to study about two major kinds of nutrition, that is autotrophic nutrition and heterotrophic nutrition. Autotrophic nutrition are of two types, that is chemosynthesis and photosynthesis. And photosynthesis we are learning in detail in this chapter. Though the textbook deals with very limited portions, because we are learning photosynthesis, we are expecting slightly here and there. But the normally the question paper does not go beyond the textbook. Okay, so you can be rest assured that whatever is covered in the textbook, if you can understand, yes, obviously, if a term is provided in the textbook and the meaning is not explained or the definition is not explained, yes, we are supposed to learn that. Okay, so to that extent, we will have to go beyond the textbook, not much beyond the textbook as far as the board exams are concerned. Whereas for competitive exams, for other this thing, we normally cover a little larger syllabus in the school also. Okay. According to the textbook, the entire process of photosynthesis happens in three steps. That is absorption of light energy by chlorophyll, conversion of light energy to chemical energy, and splitting of water molecules into hydrogen and oxygen, and reduction of carbon dioxide to carbohydrates. This has to be learned by heart as it is. No addition, no deletion. It must be learned by heart as it is. The only thing that we can do is that if you want to convert that into 4 points, because sometimes if it is asked for 2 marks in one of our exams also, we ask that key write 4 points about it. If it is asked for writing 4 points, then you can split the second point into 2 points, that is conversion of light energy into chemical energy, and second part is splitting of water molecules into hydrogen and oxygen. That much variations we can do. Other than that, stick to the textbook, use the textbook language, write as it is given in the textbook. Okay. Now, when we are talking about the first step, that is absorption of light energy by chlorophyll, if you are checking the pattern of absorption of light energy by chlorophyll, we can see light cannot, all the light available to plants cannot be absorbed by the chlorophyll. Mostly we can find that if we are going for a graph of chlorophyll A against B against carotenoids, which are another pigments available in the plants, or xanthophyll, or there are many other pigments other than chlorophyll, which can convert light energy into chemical energy. So we are going through them, then obviously we can see that in the maximum absorption happens in bluish colored light and in reddish colored light. Okay, so this graph is not given in the textbook, but this can be an application level question or maybe a paragraph type questions to check whether students are able to decipher graphs or not. Okay, there are few questions are asked like that, where the data is represented in the form of graphs. So there, this kind of a graph can be there to check whether you are able to decipher the informations related to photosynthesis from a graphical representation or not. So here we can see the light which is absorbed maximum is the bluish colored light and the reddish colored light. Whereas when it comes to green colored light, uh, normally it is not absorbed by the plant and it is reflected back by the plant. And that is why the leaves are green in color. Why? Because most of the color green colored light is being reflected by the plant. So whichever is getting reflected from the plant falls in our eyes, and we see that object in that particular color. That is why leaves are green in color. So when we are talking about absorption of light, there is a pigment system, there is a photo system, and chlorophyll molecule is the center molecule in that. Whenever light falls on the chlorophyll molecules, there are few electrons that get excited from the chlorophyll molecule, and they go to a very high energy level, and they set up a chemical reaction. And it is a very complex chemical reaction happening in the plants. In one sentence, it has been mentioned in the textbook that absorption of light energy. So what is mean by absorption of light energy? This process is what we call as the absorption of light energy, because light is carrying the energy in the form of photons, and that form of energy need to be converted into some other form which plants and animals can utilize. And that is, what is that form? And that form is the chemical form. Now, when we are talking about chemical energy, there are two different types of chemical energy. One is the chemical energy that keeps the nucleus binded together, and other one is the one that is between the nucleus and the electrons. Okay, so here the energy is getting transferred to electrons. So once the energy from the photons are given to the electrons, then we say yes, the first process has been completed. That is, absorption of light energy by the chlorophyll has been completed. Okay. Now comes the second part, that is conversion of light energy to a chemical energy. Now, electrons cannot keep the energy with them for long. If electrons are there with higher energy level, they will indulge. First and foremost thing that is going to happen is that they will leave the atom and run away, because when they are getting more energy level, they will go to the larger orbits. They will move away from the nucleus because they have more energy, and they will start revolving in a larger orbit. And sometimes it can get to such a distance from the nucleus that they will leave the atom and will start roaming somewhere else. And another atom which can accommodate this electron will absorb them and will start utilizing the energy for some chemical purposes. That means electrons with very high energy level in any system can set off as can trigger a reaction. So a lot of chemical reactions start, and at the end of the chemical reaction, you get some chemical. So there are two steps happening. One is absorption of light energy into chemical energy. Absorption of light energy by the chlorophyll molecule, that is exactly mean by the conversion of the energy of the photon into the energy of electrons. And in chapter number 15, we have studied that how much amount of energy can be absorbed, or what is the rate of this conversion. What is the rate of this conversion? It's only 1%. If you remember, we studied the 10% law in chapter number environment, and the number has changed in the new book. Sorry, in chapter environment, our environment chapter, we studied the 10% law. According to that 10% law, when energy is flowing from one trophic level to the next trophic level, only 10% is transferred to the next trophic level. But the percentage of absorption of energy from the sun to the plant, or the amount of energy absorbed by the plant, is just 1%. And what is that 1%? That 1% is this conversion rate. Which conversion rate? Absorption of light energy from the photon to electron. Okay, so out of the total amount of energy that falls on a chlorophyll molecule, only 1% can be converted or can be given to the electrons, can be converted into the energy of the electrons. The rest all are uh, uh, going wasted. Means plants cannot use it. It will be there in the nature somewhere, but plants cannot use it. Once the energy is given to the electrons set off a chemical reaction, and they end up in making some chemicals. There are many chemicals which we have discussed in the class also, or you must have heard in previous thing also. that NADPH or ATP, these kinds of molecules which can store energy for some time and can release it whenever it is required. So it's a kind of a reversible chemical reaction where you can charge and discharge. So there are certain chemicals like FAD, flavin adenine dinucleotide, or NAD, nicotinamide adenine dinucleotide, or phosphorylated form of that, or reduced and phosphorylated, that is NADPH. Or you can go for ATP, which is a very commonly known term, adenosine triphosphate, where with addition of every phosphatic molecule onto adenosine molecule, energy is getting added onto that, which can easily be released. Okay, there are some elements which take up the energy and get undergoes a chemical reaction and convert to some other chemicals, but it should be something that can be reversed also. So these kinds of chemicals like NADPH2 or FAD or ATP, these kinds of molecules are interconvertible. So once the energy is reaching there, or the energy absorbed by the um, electron is being used for making that, we can say the energy of the light has been absorbed. Step number one, energy of the light has been converted into light energy, or say chemical energy. Once it has been done, the energy is also being used for splitting a water molecule. Okay, now uh, splitting a water molecule is very much essential because uh, uh, oxidation, sorry, photosynthesis is not an oxidation process. It's a reducing process. It's a reduction reaction. Why is it? Why can we say that it is an it is not an oxidation process? Because in most cases, if you are finding oxidation processes are destructive processes. They break down the material into smaller compounds, whereas reduction reactions can build up uh, molecules. So from smaller elements, compounds like water and carbon dioxide, we are getting a larger compound as a kind of a combination reaction. And so it is a reducing reaction. So for reducing carbon dioxide into carbohydrate, we need hydrogen. And that hydrogen is actually released from the water. And breaking of water molecule needs some energy. And that energy is also provided by the sun. Okay. So here is a figurative representation of the same, how the energy from the sun is being used for absorption of light energy and breaking down of water into hydrogen and oxygen. And this hydrogen will be added onto carbon dioxide, or it will be used for reducing carbon dioxide to carbohydrate. So these are the chemical that I was discussing about. Let me go quickly. The third step involved in photosynthesis is the reduction of carbon dioxide to carbohydrates. Reduction of carbon dioxide to carbohydrate. That means addition of carbon, so addition of hydrogen into carbon. One of the chemical that is helping in this entire process is rubisco. Rubisco. It has got many names like ribulose biphosphate, ribulose 1,5 biphosphate, or rubisco, or these kinds of terms are used for this chemical. This is actually an enzyme which can speed up the process. What process? It can speed up the entire process of uh, conversion of or utilization of NADP or ATP to add hydrogen into carbon. That entire process is actually happening in the form of Calvin cycle. We are not studying Calvin cycle in detail in class 10, but only the name would be sufficient. So there are two major activities happening in the process of photosynthesis. One is absorption of light energy by the chlorophyll and storing or converting that into some chemical form. And what is that chemical form? It's either ATP or NADPH. It's actually a kind of stored form. And from there, it will be utilized for the reduction of carbon dioxide, which can be done at any time. For example, it is not necessary that it should be done or utilized the same time or not. It depends on the type. One of the example given in the textbook is about desert plants. During that time, they get the sunlight. And at that time, they make a lot of ATPs and NADPH and then store it in the plant. If we are going for the second step, then there is a danger. And what is the danger? They will have to open up their stomata for gaseous exchange. Because carbon dioxide, water when split into hydrogen and oxygen, the oxygen has to be released out. There is no adequate space inside the plant to store so much of oxygen. So they will have to open the stomata for releasing out oxygen. If they are opening the stomata along with oxygen, a lot of water will also be escaping. So in order to avoid that, they don't do that during daytime. They store the energy in the form of ATP or NADPH, and then they wait for the night time. And during night time, they start the second phase of the chemical reaction. So because the first phase depends on light, we call it as it's a light dependent phase, or in short, we say that it's a light phase. And the second step does not require light, so we consider it as a light independent phase, or we, in short, we say that it's a dark phase. But while referring it to as dark phase, it does not mean that it happens only in dark. And it simply means that it is a light independent phase. Okay, it may happen during daytime or it may happen during nighttime also. That depends on how much amount of energy the plant has stored in it, right? So this is the third step and the final step, where the carbon dioxide will be taken inside, and the water will be split to release hydrogen from it, and complex chemical reactions in the presence of Rubisco converts carbon dioxide plus hydrogen into glucose. Okay, and that glucose can be stored in the plant, but glucose cannot be stored in the plant, and the glucose subsequently will be converted into starch, and the starch will be stored in the plant. Okay, I'm going a little fast because I have limited time. A quick revision only. Okay, so if we are asking about one of what is the comparison between light reaction and dark reaction, probably this can help you. When anybody wants to take a screenshot, can take on and then can ask me questions later. Some points are beyond the textbook, so you can avoid that. If we are writing, that's also absolutely fine. Let me jump over to the next one. The next portion in the textbook talks about the factors affecting photosynthesis. That majorly includes four major factors: chlorophyll, carbon dioxide, water, and sunlight. And there are some activities related to that. Earlier, we used to do it in lab. There are some activities related to that, and some the practical based questions were there in the question paper, that is annual theory paper. But since last few years, that pattern has been changed. But this year is one of the things, extra questions that contains one question related to the activity. So we are expecting that there will be some questions related to activities related to these factors affecting photosynthesis also. So some of the very common activities are: number one, this one. How can we test the presence of light is essential for photosynthesis? So this is a very commonly done activity, where we are taking a leaf and then covering small portion. But before doing this, we must de-starch the leaf. I hope you are aware about what is meant by de-starch. Removing the starch from the plant, which is already there in the plant. So keep it in dark room for few one day or so, and then take it out, so that we can ensure that there is no or pre-prepared starch on leaf. And then we can cover some part of the leaf with that black pepper and cover it properly, and then place it in the sun, and later we can go for an iodine test. But iodine test on a green leaf is not visible, so we need to decolorize the leaf before doing this. Decolorizing can be done by boiling in water and then boiling in alcohol, right? So alcohol can dissolve away the chlorophyll, and the alcohol becomes green in color, and leaf becomes almost transparent, translucent, or it loses the green color, we may say. Okay, and then we can go for an iodine test, and it is expected that other than the covered portion, rest of the portion should convert into green to blue black, showing that the other portions photosynthesis has happened and glucose must have formed, and that glucose must have converted into starch. Okay, this is another one explaining chlorophyll is essential for photosynthesis, where we are taking a variegated leaf. A variegated leaf is a leaf that is having a different patches. We prefer a leaf that is having green and white patches, and then doing the same experiment, discharging, then putting it in the sunlight for few hours, and then decolorizing, and then doing the starch test on it. So there also, it is expected that the yellow colored area or the white colored area should not give you any positive result, but the green colored area should give you a positive test for the starch test. That is, it should convert into blue black, showing that the green portions normally have chloroplast and they can do photosynthesis, and that can produce starch. Otherwise, it is difficult for us to test the presence of glucose. Hmm. Yes. There is another experiment that is called very often asked, that where we are testing that carbon dioxide is essential for photosynthesis. For doing that, we need to grow the plants in an atmosphere devoid of carbon dioxide. At the same time, we should take care that all the other components are provided. For that, we are making use of some chemicals which can absorb carbon dioxide very fast. Any kind of hydroxide can do this work. Here we are using potassium hydroxide. If you don't have potassium hydroxide, you can also make use of calcium hydroxide. That is also easily available. Calcium hydroxide is also available. So that can also be used. Either potassium hydroxide or calcium hydroxide can be placed on the side of a potted plant and can cover it with a bell jar. A bell jar normally covers it very tightly. It does not have any opening on the top. At the same time, its bottom is completely sealed, ensuring that no exchange of gases happens between the bell jar and on the inner surface, inner side and the outer side. And because of that, we are ensuring that this plant is growing in a carbon dioxide free atmosphere. We are placing it inside for two purposes. Number one, it will absorb away the carbon dioxide which is already present inside the jar and will create a carbon dioxide free environment. At the same time, if the leaf is doing photosynthesis, during photosynthesis, during if it is undergoing respiration, it will release carbon dioxide. That carbon dioxide will also be absorbed away by the potassium hydroxide or calcium hydroxide. So that way we can ensure that it is living in an atmosphere without carbon dioxide. Then we can go for the regular decolorizing of the leaf and then starch test. Okay. So these are the factors. Other than that, the two other factors which are affecting water and sunlight, there are no activities given in the textbook for testing this. There is one more activity given that is related to respiration, but that I will show you after with the portion respiration. So this is how the nutrition happens in plants. Okay, keep your doubts noted, and then we shall ask together. Okay, and let me quickly jump over to the next portion that is included in this chapter. That is nutrition, heterotrophic nutrition. Yes. The second part of this is heterotrophic nutrition, where the organism depend on another organism for their nutrition. Okay, that majorly includes three major category types. One is saprophytic nutrition, parasitic nutrition, and holozoic nutrition. All three are subtypes of heterotrophic nutrition. Organisms may go for saprophytic, parasitic, or holozoic, but take care that it is not always compulsory that the organism which are performing saprophytic or parasitic nutrition should be always animals. They can be plant also. So, for example, cuscuta. Okay, the spelling is printed wrong. Sorry. Yes, cuscuta. You must be aware about this one. CUSCU. Cuscuta. Yes, is a plant. But what is its mode of nutrition? It's a complete parasite plant. Right. So we are not categorizing organisms whether they are, or we are not separating organisms whether they are plant or animal. We are discussing about the type of nutrition majorly. So if an organism is depending on another organism for nutrition, whether it is a plant or animal, yes, you will categorize them into these three categories. Number one, if an organism is digesting their food outside their body, if maybe due to any reasons, either they do not have any digestive system at all. For example, in case of fungus, they don't have any digestive system. Their cells are incapable of digesting food, but they do create, they do make digestive enzymes. These digestive enzymes are normally poured outside, and the food is digested outside, and they absorb only the nutrients. Then that can also be considered as the saprophytic nutrition. Okay, bacteria, fungus, there are some organisms, some varieties of spider, they are also showing saprophytic mode of nutrition where the food is digested outside the body. Hmm. Saprophytes are different from scavengers. The organisms which are depending on dead and decaying material are not all of them are not saprophytes. We normally say that okay, the organisms that depend on dead and decaying material are saprophytes. But when it comes to say scavengers, they do eat dead and dead organisms like a hyena or a vulture. What is their major food? Their food is dead plant animals, but they do eat it. They have a well developed digestive system. The food goes into the digestive system and they get that get digested, just like any other organism. So they are not saprophytes, they are holozoic organisms. Parasitic nutrition is when two organisms live together and one of them is benefited at the cost of another one. Okay, one is getting food from another one and the other one should be harmed. It's not only that only one is getting benefited. At the cost of the harm goes to another one. And then we can say that yes, that's a parasitic nutrition. Right? And holozoic nutrition, the organisms which consume the entire food, they mostly have a very well developed digestive system, and the food is taken into the digestive system and digest inside the digestive system. Okay, it can be a complete digestive system or an incomplete digestive system. A complete digestive system has got two openings, one for the entry of the food and second for the exit of the undigested food material. Whereas in smaller organisms, the digestive system is not very well advanced. So they have only one opening. The food enters into the body through that opening, and they may have a very primitive kind of a stomach or intestine or something like that. We can't even call it as an intestine. We can better call it as a body cavity or something where the food get digested. And undigested food materials are thrown out through the same opening. Okay, so that is another way of, we can say that incomplete digestive system. Okay, so these organisms, the holozoic organisms normally have any one of these kinds of digestive system, and they take the food into their body. It may be cut into pieces, it may be chewed, it may be swallowed as it is, but the question is that whether the food is entering into the body and digesting inside the body or not. If it is happening, yes, they are holozoic nutrition. These are saprophytes, some parasites, and holozoic. When it comes to holozoic nutrition, it follows five steps, whether it is human being or any other organism. It follows 5 steps: ingestion, digestion, absorption, assimilation, and egestion. Ingestion happens when the food is taken into the body. Different organism uses different methods for ingesting the food. Digestion is the chemical breaking down of food. Digestion can be further divided into two parts. One is the physical digestion and the other one is chemical digestion. Physical digestion includes when larger particles are broken into smaller particles. Absorption happens inside the body where the digested food materials are absorbed, and assimilation is not happening in the digestive system. It is happening across the body or throughout the body, where the digested food material will be consumed or utilized for some or the other activities of the body, for the building a process of the body, or some it may be converted into a part of the body, or it can be consumed as in the form of energy, and that process is what we call as assimilation. Digestion happens in either in the stomach or buccal cavity in case of human being, or in some part of the digestive part of the organism. The absorption also happens there. If it is a unicellular organism, the absorption happens directly into the cytoplasm. If it is a multicellular organism, they may have some systems where the absorption can be done, and whereas assimilation is happening in each and every cell of the body. Okay, and then there will be something called egestion, that is removal of the waste material. Now, this egestion is not equivalent to excretion. In the other topic, we are studying about excretion, where the physiological wastes are released, whereas this is egestion. Egestion is removal of undigested food material, and these are not the creation of the organism. These are the material that came from outside, but the organism cannot utilize it, so it is going out. Okay, one example of that is amoeba. There is a four-step process explained in your textbook. This diagram is quite often asked, so go through this diagram. Okay, you can increase the number of this thing also. Okay, now let me quickly jump to the next portion of that, that is human digestive system. Human is a holozoic animal. Human digestive system is a very complex digestive system. It has got many parts involved, so we need to study them in detail, but I'm going a little quickly. The first portion is the mouth and the buccal cavity. All the components of the mouth and buccal cavity need to be learned. We have at least three different organs in the buccal cavity. One is the teeth, the tongue, and the salivary glands. Teeth is helping in physical digestion. Tongue is assisting it in both the physical and chemical digestion, and salivary glands start the chemical digestion. Salivary glands can produces saliva. Okay. Teeth and this thing, and then comes saliva. Salivary glands. There are three pairs of salivary glands in the mouth or in the buccal cavity, and they produce a chemical called the saliva, and saliva contains some chemicals. It contains many chemicals in it. It contains lysozymes for killing the bacteria. It contains amylase. One of the most important thing is amylase or thialin. It has got another name also, not very often used, but if it is appearing in the question paper, you should be aware about it. Thialin. Thialin is the word used for salivary amylase. P is silent here. Thialin. So this is the first digestion happening in the body, that is, which converts starch into sugars. Starch into sugar, most probably into maltose, and this is the first digestion that happens. Starch can be digested at different parts of the body. One important part is the mouth. So the moment you place the food in the mouth, the digestion starts. So in human body, the digestion starts in the mouth. Okay. There is an activity also related to check the effect of salivary amylase on starch, where we are placing two test tubes with starch in it, and one is with amylase, one is without amylase, and then going for the starch test. The one without amylase should not will give the starch test, whereas the one with amylase will not give the starch test. Why? Because amylase have converted all the starch into sugars. The lower portion of that is what we call the esophagus, which is filled with muscles, smooth muscles, involuntary muscles, and they show rhythmic contraction and expansion, and which is called as peristalsis. This peristalsis is not only found in the esophagus, it is starting from the esophagus, but it is extending all the way to the large intestine also, to the rectum also. Okay, this is the movement that is pushing the food continuously forward. The largest effect of or the maximum amount of peristalsis can be found in the stomach, where we call the stomach is churning the food. That's actually a kind of peristaltic movement. So with the help of this peristaltic movement, we can see the food. After mixing it up with saliva in the mouth, we don't call it as food because the name keeps on changing as it changes its shapes and its um, combinations. Once it is mixed with saliva, then it is called as bolus, bolus, bolus. The bolus enters into the esophagus and goes all the way into the stomach. At the entry of the stomach, there are certain muscles arranged which opens only when the food goes inside. It cannot open outward, and they are made up of very strong muscles called the sphincter muscles. The upper esophageal sphincter muscles. Sphincter muscles are placed at many places in the digestive system. Wherever we need to stop the movement of material, the default position of a sphincter muscle is tightly closed. You need to spend energy to open it. Otherwise, it remains tightly closed. Okay. Then the food reaches the stomach. The stomach produces three major chemicals. It has got gastric glands on the inner side, and that produces gastric juice. Gastric juice contains mucus, hydrochloric acid, and pepsinogen. Okay. The functions of mucus is protecting the inner walls because very dangerous chemical reactions are going to happen. It contains hydrochloric acid, it contains pepsin, which is a very strong protein digesting enzyme, which is called proteolytic enzyme. And to protect it from the high effect of that, we have a coating of mucus there. Then hydrochloric acid mainly kills the bacteria and creates an acidic medium. The chemical reactions, it is good to remember if it is possible, because it is not much detail in the textbook. Normally, proteins are digested by pepsin present in the gastric juice, and it will be broken down into smaller components called the proteases and peptones. These proteases and peptones enter into the next level, that is the duodenum, the small intestine. Small intestine is divided for study purpose into three categories: duodenum, jejunum, and ileum. The duodenum portion, which is the curve just after the stomach, and that curve, there are two major organs connected. One is the liver, and the other one is the pancreas. Both of them, the tubular outlet from both of them join together and enters into the duodenum with a single opening, that is called as the common bile duct. Liver produces a chemical called the bile, and it is stored in the gallbladder for some time. Bile does not contain any enzymes, but it has some chemicals which can do some chemical reactions. Now, what is the difference between an enzyme and a reagent? A reagent, the bile juice contains reagents. Normally the reagents react with a chemical and get converted into something else, whereas enzymes are biocatalysts. Catalyst is something that can perform a chemical reaction, and at the end of the chemical reaction, the catalyst is free. The catalyst can change the speed of the chemical reaction, but it does not get used up in the chemical reaction. So enzymes are like that. Enzymes can be reused. But in human body, what happens? The enzymes get moved out with the food forward into the next level. So it may not be useful. But Enzymes chemically can be reused. And bile juice does not produce any enzymes, whereas pancreas do produce some enzymes. Okay. And the bile juice normally produce bile, and their major function is breaking down of larger particles of fat or oil droplets into smaller particles. And this is called the uh, this thing, emulsification of fat. Okay. Once it has been converted into smaller parts, the emulsification has done, that increases the surface area. So the next set of chemicals can act on it, and that is the bile pigments. Two major bile pigments I have mentioned here. One is bilirubin, biliverdin. Not mentioned in the textbook, but if it is somewhere asked, or if it is available in any of the MCQ or somewhere, you should be aware about it. And these chemicals helps in the next process, that is saponification, conversion of a water insoluble material into a water soluble material. This is the same chemical reaction that we are learning in chapter number 4, carbon and its compounds under saponification, where we are using either soaps or detergents to get rid of dirt from cloth. The chemical reactions are almost similar. There also, we are converting the soap or detergent is making a missile. What is that? Water insoluble dirt will be covered in soaps or detergent, and then it becomes a water soluble material. The same thing is happening in the small intestine also. The water insoluble fat molecule will be converted into a water soluble material, because human being has a water based system, that is the blood and the lymph. If it has to be imported or taken into the blood or lymph, it must be water soluble. So bile pigments, they help in this process of conversion of bile water insoluble material to water soluble material. Okay. So that is how they are helping us. Then the next major chemical that is released into that region is the pancreatic juice. Pancreatic juice again helps in the digestion of proteins. Pancreas is a dual gland which produces hormones also, enzymes also. Pancreatic juice contains many chemicals in it. One of the major one is the trypsin, which further divides or breaks down proteins into further smaller compounds or protease and peptones. Trypsin breaks down polypeptides or dipeptides into proteases and peptones into dipeptides. So that will help in breaking down the protein molecule further. And then the food moves forward into the intestinal area, where the intestinal juice is produced. Intestinal juice contains many chemicals for digestion of almost all the components, like the peptidase, sucrase, maltase, lactase. All these chemicals are produced in the intestinal wall, and they complete the digestion in the small intestine. Okay, and so digestion starts in the mouth and buccal cavity, and then ends in the small intestine. So this is another representation how the three different organs are helping in breaking down large protein molecule into very simple amino acids. It happens in three different steps. One is pepsin in stomach converts proteins into polypeptides. Then trypsin in duodenum converts polypeptides into peptides or dipeptides. And in small intestine, the intestinal juice are converting the peptides into amino acids. So in three different steps, the digestion of protein completes. And then comes the last portion, later part of the small intestine, where on the inner side we can find that the inner lining is covered with many tiny tubular structures, which are called as a villi. They help in absorption of the digested food materials into the stomach, into the bloodstream. Each villi is provided with a large amount of blood vessels and the lymphatic vessels. There are many material which can be taken into the blood vessels, whereas some larger molecules cannot be taken into the blood vessel. They will be transported through the lymphatic vessels. Lymphatic system is also another system which is present in the stomach, in the body, which helps in transport of material. The undigested food material normally enters into the later part, that is the large intestine. And here, majorly the absorption of water and salts happen. And large intestine is a home for many bacteria, symbiotic bacteria also. And these bacteria help in making a large amount of vitamin B complex. At the end of the large intestine is a small bowl-like structure called the rectum, and rectum collects the undigested waste material for some time. Okay, so these are some of the major portions involved in heterotrophic nutrition. Again, I would like to stop here for the day, and we welcome some questions if you have any.